Hello everyone, welcome back to my channel. In this session I will be actually continuing with the development of nervous system. So those who haven't watched my session on development of nervous system, please do watch that session first, then continue with this topic. Okay, so in the last session on the development of nervous system, we have seen the formation of neural tube and the formation of neural crest cells. Okay, so now we are going to deal exclusively with the neural crest cells because it is one of the favorite questions for again university exams as well as for the entrance question. So we'll be seeing in detail about the neural crest cells because as a joke we usually say whenever someone asks some particular cells derived in the body, you can simply say that it is from the neural crest cells because that is the extent of cells which are derived from the neural crest cells. That is a simple joke. So we will be seeing it. So first what we have seen is you have the ectoderm here and you have the neural tube here, you have the notochord here. So at first it will be just a wedge shaped mass between the ectoderm and the neural tube. And finally what happens is it will just divide into two and it will migrate on either side of the neural tube. So this is actually the dorsal aspect. So this mass will be lying first on the dorsolateral aspect of the neural tube. So this will just separate into two parts and they will be lying on either side of the neural tube on the dorsolateral aspect. This is the ventral aspect, this is the dorsal aspect, and so dorsolateral aspect. Then as soon as these oval masses are formed, they will again split into two. Okay, so you can see a mass at the dorsal aspect and you can see a mass in the ventral aspect. So this is altogether known as primitive ganglia. Primitive spinal ganglia. Okay, so these two masses, the dorsal mass and the ventral mass together forms the primitive spinal ganglia. Now we will see the derivatives of this dorsal mass as well as the derivatives of the ventral mass. So first you have a wedge shaped mass between the ectoderm and the neural tube which splits into two and lies on the dorsolateral aspect of the neural tube. Then again each mass on either side will again split into a dorsal mass and a ventral mass. And now we will be seeing the derivatives of the dorsal as well as ventral mass. So generally speaking, for easy remembrance we usually say that the dorsal mass is giving rise to the sensory ganglia whereas the ventral mass is actually giving rise to the autonomic ganglia, the sympathetic and parasympathetic ganglia. That is what we roughly say. So first we will focus on the dorsal mass. So this is the dorsal mass. And the dorsal mass, as we have already seen, it is giving rise to the dorsal root ganglia of the spinal nerve as well as the sensory ganglia of the cranial nerves, that is 5, 7, 9 and 10. So these are the ganglia which are mainly formed from the dorsal mass. Now there are mainly three types of cells formed in this dorsal mass. We can call it as neuroblast cells, pluripotent cells and the spongioblast. Neuroblast, pluripotent and the spongioblast. So neuroblast cells are actually first of all in the shape of an oval. Okay, so it is oval in shape in the beginning and it has got a central process and a peripheral process. So this is how a neuroblast cell will look like in the beginning. Then this will actually go and merge with the dorsal aspect of the neural tube, the central process, and the peripheral process which will act. So this central process will be actually forming the sensory root of the spinal nerve, and the peripheral process will actually merge with the ventral root and will end in the sensory end organ. So that is our neuroblast cell. So it will be first oval in shape with a central process and a peripheral process. Central process will go to the dorsal aspect of the neural tube and it will form the sensory root of the nerve, spinal nerve, and the peripheral process will actually join with the ventral root of the spinal nerve and it will end in the sensory end organ. Now what happens is these two processes, the central process and peripheral process, will try to come closer. So what happens? You have a cell with the two process fused. Okay, and then you have the peripheral process and you have the central process. So this is actually in the form of a T-shaped bipolar neuron. So that is the peculiarity of this. So it will first look like a unipolar, but it is not unipolar. We know that it was a bipolar neuron which was made into a unipolar neuron. Hence it is known as pseudo unipolar neuron. Okay, so the central process and peripheral process will actually come in the midline and they will fuse, and from this single point it will just go to either side. So we will feel like it is just formed from the cell and it is splitting. So it's not like that. According to the development, you have an oval cell with a central process and a peripheral process. So these type of pseudo unipolar neurons are seen in the dorsal root ganglia as well as the sensory ganglia of the cranial nerves. But this bipolar neurons persist in certain regions like retina and vestibulocochlear cells. So in those regions we still have the bipolar cells, but whereas in the dorsal root ganglia and the sensory ganglia of the cranial nerves we have the pseudo unipolar neurons. So pseudo unipolar neurons are formed in this way. Now we will come to the pluripotent cells. Pluripotent cells, we know that they can give rise to any type of cells. So which are the types of cells which are formed from these group of cells? We have the cartilage cells of the branchial arches, we have the odontoblasts of the developing teeth, then you have the melanoblast, that is the pigmented cell, and you have the parafollicular C cells of the thyroid gland. So these are the different types of cells formed from the pluripotent cells in the dorsal mass of the primitive spinal ganglia. Now the third group of cells which you will get in the dorsal mass is spongioblast. So what are they responsible for? What are the cells formed from the spongioblast? We have mainly two types of cells formed from the spongioblast. The first one you call it as capsular cells, and the second one you call it as Schwann cells. So where will you get the capsular cells? Capsular cells will be actually arranged around this pseudo-unipolar neurons. Okay, so the cell body of the neurons will be surrounded by the capsular cells. And where will you get the Schwann cells? The Schwann cells will be, if you have the spinal cord like this and if you have some naked axons emerging from the spinal cord, these Schwann cells will go and align along the nerve. And finally what happens? It will actually wind the axon in a spiral manner and it will result in the deposition of myelin. So myelin is actually alternating protein and lipid bands. So you have the formation of myelin sheath from the Schwann cells. So if you take this as the peripheral nerve, you have the Schwann cell in the form of rotating loops, and towards the periphery you have the nucleus of the Schwann cell. So you have the myelin deposition in between the layers of the Schwann cell, and the outermost layer is what is known as neurolemma sheath. Neurolemma. Okay, so the outermost cytoplasm along with the membrane, you call it as neurolemma sheath. So the Schwann cell will give rise to two entities. One, the myelin sheath of the myelinated nerves, and the outermost covering is known as neurolemma sheath. Neurolemma sheath. So we know that myelin sheath is actually making the conduction faster, and so there are many unmyelinated nerve fibers, right? So what happens in the unmyelinated nerve fibers? What is the role of Schwann cell there? The Schwann cells there will be giving rise to the formation of neurolemma sheath. There won't be any myelin formation, but still, since the Schwann cell is surrounding the axon, you will get the neurolemma sheath. In myelinated nerve fibers, you have the myelin sheath inner to the neurolemma sheath, and the outermost is actually formed by the neurolemma. So that's about the derivatives of the spongioblast. So we have seen the three different groups of cells: neuroblast, pluripotent cells, and the spongioblast. So I've tried to make a code for this to remember. So if you find it easy, please leave a comment. So NB we usually write it as a code for please note, right? So NB, Please come Paul, C caps. Okay, so just focus on the capital letters. So capital letters will give the derivative. So NB means neuroblast and the bipolar cells which are derived from it. Please come Paul. So please, it is the capital P. So that represents the pluripotent cells. Then come, it is only COM, what I meant. So COM stands for the cartilage cells, odontoblast and the melanoblast. And the P of the Paul stands for parafollicular C cells. And the next one is C caps. So C, the S stands for the spongioblast and caps C and S. Cap is actually the capsular cell and S is the Schwann cell. So NB, please come for C caps. So that will give rise to the derivatives of the dorsal mass of the primitive spinal ganglia. Now we will move on to the ventral mass. So these are the ventral masses. So the ventral mass is actually they are just traveling across the body to different regions, and they are actually forming at first the sympathochromaffin organ. So altogether, this is a group of nuclei. So you call it as a syncytial mass. So this syncytial mass was at first known as the sympathochromaffin organ. So the sympathochromaffin organ has got two sets of cells. Some are small and some are large. So the small cells are actually the sympathoblast cells. Okay, the small cells of the sympathochromaffin organ, you call it as sympathoblast cells. And what are derived from the sympathoblast cells? Sympathoblast cells are actually giving rise to the sympathetic ganglia and the parasympathetic ganglia. Okay, so first these cells will go on either side of the vertebral column to reach the dorsal aorta. So primitively speaking, we have two dorsal aortae. So if you have the two dorsal aortae like this, these cells from the neural crest, the ventral mass of the neural crest cells will migrate and will lie along the dorsal aortae. And each cell will be having many processes, isn't it? So these will get interconnected. So these cells will get interconnected, and finally what happens? You will get the vertebral column in the midline, right? So on either side of the vertebral column, you will get a chain of ganglia. So that is our sympathetic chain. So that is how the sympathetic chain is formed. So the small cells from the ventral mass will migrate on either side of the vertebral column and will lie along the dorsal aortae, and they will be having many processes, and these processes will get united and ultimately they will form the sympathetic chain on either side of the vertebral column. Now you might get some of the cells traveling along some of the cranial nerves to form the parasympathetic ganglia. So let's see which are the parasympathetic ganglia seen in relation with the cranial nerves. So third cranial nerve, you have the ciliary ganglion. Then the seventh cranial nerve, you have the pterygopalatine ganglia and the submandibular ganglion. Then you have the ninth cranial nerve, which is related to the otic ganglia, and you have the 10th cranial nerve, the vagus. Vagus is actually parasympathetic to the gut, right? So they will be forming ganglia near the target organs. So these are the cranial nerves, and the cells from this ventral mass, the small cells from this ventral mass will be traveling along these cranial nerves to form the parasympathetic ganglia. Likewise, the third in relation with the ciliary ganglia, 7 will be seen in relation with the pterygopalatine and submandibular ganglia, 9th in relation with the otic ganglia, and the 10th at the end organs, the target organs of the GIT. Now the next group, the larger cells, so smaller cells are the sympathoblast, and the larger cells of the sympathochromaffin organ, you call it as chromaffin cells. So chromaffin cells are called so because they have brownish granules which are stained with chromic salt. So that is why they are known as chromaffin cells. So chromaffin cells, where are they seen? A major portion of these chromaffin cells will go to the adrenal medulla. We know that the suprarenal glands or the adrenal glands, you can see a session on the development of adrenal glands, I have done it. So they have a cortex and a medulla. So these cells, the chromaffin cells, will be moving towards the medulla in order to form noradrenaline. So it is noradrenergic. But in human beings, what happens is you have the cortex, and you have the medulla towards the inner aspect. Since the medulla is lying in close relation with the adrenal cortex, what happens is the noradrenaline formed in the adrenal medulla will be easily converted into adrenaline by the methylation of the amines. Okay, so ultimately what happens is the medulla will be secreting more adrenaline, though it is not adrenergic. Now another group of cells, you call it as argentaffin cells. So what are the argentaffin cells? They are the chromaffin cells seen in other regions, like on the surface in relation with the mucous membranes of the respiratory and GIT. So all these cells seen in relation with these regions, you call it as argentaffin cells. And the cells which are seen in close relation with the GIT, they are given a particular name that is known as enterochromaffin cells. Enterochromaffin cells. So enterochromaffin cells are the chromaffin cells seen in relation with the mucous membrane of the GIT. And they are peculiar for the production of some local polypeptides, and hence they are known as Apert cells, amine precursor uptake and decarboxylation, Apert cells. So that is argentaffin cells or enterochromaffin cells. The next place where these chromaffin cells are deposited is known as paraganglia. Paraganglia means these group of cells will be located in the sympathetic ganglia. We have already mentioned about the sympathetic chain, right? So you have the sympathetic ganglia like this. In the sympathetic ganglia, these chromaffin cells will go and get deposited to form the paraganglia, and they are actually dopaminergic. They will be producing dopamine, and they are also known as small intensely fluorescent, SIF, small intensely fluorescent cells. They are the paraganglia. And another place where you have the chromaffin cells seen are known as para-aortic bodies. So para-aortic bodies, where will you look for the para-aortic bodies? So we know that we have the aorta, and we have three main arteries in the abdomen arising from the aorta, isn't it? Celiac trunk, superior mesenteric artery, and the inferior mesenteric artery. So the para-aortic bodies are actually the chromaffin cells which are seen in the form of inverted U-shaped loop, okay, along the inferior mesenteric artery. So the chromaffin cells will migrate onto the aorta, and at the place of origin of inferior mesenteric artery, they will get deposited in the form of inverted U. They are known as para-aortic bodies, and actually they are also noradrenergic, because the chromaffin cells usually are known as noradrenergic cells. So these are the derivatives of the chromaffin cells. So if you just consider the starting letters, you can just call it as MAP. You can make a beautiful map using the chroma means color, right? So you can make a beautiful map using the colors. So MAP, M stands for medulla, the adrenal medulla, A stands for the argentaffin cells, P for the paraganglia, and another P for the para-aortic bodies. So these are the derivatives of the ventral mass. And I forgot to mention one thing, where will you see the neural crest cells? So which all location? So you have the neural tube starting from the cranial end towards the caudal end, right? So you have the neural tube like this from the cranial end towards the caudal end. In the cranial end there are three brain vesicles, and the caudal end will remain as a tube, and it will be forming the spinal cord. We will be seeing in details of the brain vesicles and all. So forebrain, midbrain and hindbrain vesicles, the prosencephalon, mesencephalon and rhombencephalon. Then you have the spinal cord. So these neural crest cells will be seen on either side of the neural tube from the midbrain vesicle till the caudal end of the neural tube. So this is the location where you have to look for the neural crest cells, from the midbrain vesicle till the caudal end of the neural tube. You have the neural crest cells. So just to wind up the session, we have the neural crest cells which are first formed from the neuroectoderm, neural folds of the neuroectoderm. They are not actually going and joining with the neural tube. They remain as a separate mass, first in the middle, then they split into two, and each mass will again split into a dorsal mass and a ventral mass, and they are together known as primitive spinal ganglia. Later the dorsal mass will be giving rise to the sensory ganglia, the dorsal root ganglia and the sensory ganglia of the cranial nerves, 5, 7, 9 and 10, and the 3 groups of cells formed are neuroblast, pluripotent and spongioblast, and the ventral mass is together known as sympathochromaffin organs, which has 2 derivative cells, cell groups, the small cells and the large cells. Small cells are known as sympathoblast, which is mainly giving rise to the sympathetic chain and the parasympathetic ganglia, and the chromaffin cells will be giving rise to the cells of medulla, the argentophine cells, the paraganglia and the paralytic bodies. That's all about the neural crest cells in a nutshell. In the coming sessions, we will be seeing in detail about the development of brain vesicles. So before that, I would like to take a session on the interior of spinal cord, because that is very important in order to understand the development as such. So in the coming sessions, I will be dealing with the interior of spinal cord and some of the important topics which you have asked me. So keep watching. Those who haven't subscribed, please do subscribe so that I will get more motivated to do more videos. Thank you. Thanks for watching.